So to understand the basics and fundamentals of biology, you first have to start with chemistry. Chemistry is a physical science, but all things in biology, in the function of life, depend on these chemical reactions, which are based on molecular and atomic interactions within living things. So we're going to start at the very basic level and then build from there. So the first off, the question that's most basic is, what is matter? Well, matter has mass, okay, by definition. This is a little bit different than weight. Weight is a measurement of gravitational pull. Mass is something that occupies space, okay, and is composed of atoms. Atoms are our basic element of matter. Now it does get smaller than that, but for the purposes of this class, that's where we're going to start. Now atoms themselves are made of subatomic particles, and the properties of these subatomic particles determine the nature of that atom. There are three subatomic particles: protons, which have a positive charge, are located in the nucleus, and are have a mass of 1 atomic mass unit, which is what AMU stands for. Neutrons do not have a charge, but they are also found in the nucleus and also have a mass of 1 atomic mass unit. And then finally, electrons. These orbit the nucleus. You can see that in the yellow circular pattern there around our central nucleus. They do not have a mass. Well, they do have a mass, but it's so negligible compared to the protons and neutrons, we're going to say it has a relative mass of 0. It has a negative charge, and so it has a charge which is the opposite of the protons and can balance protons in a 1-for-1 ratio. Okay. All right. These subatomic particles, like I mentioned before, determine the chemical properties and the identity of atoms. These atoms, which are different, are called elements. Elements are determined or defined by the number of protons in the nucleus. So this also corresponds with their atomic number. You can see the little hydrogen there in the middle, in the key. The number above the atomic symbol, or the chemical symbol, is the atomic number. And each of these atoms or elements has by definition, another chemical symbol. So beryllium is the atomic number 4. It has a chemical symbol Be, and it has 4 protons. So that's also what that 4 means. All right, now if protons determine what atom it is or what element it is, the other subatomic particles in the atom determine other properties, including the charge, mass. So it is possible to have atoms of the same kind that have a different mass. And so example we have here are carbon-12, carbon-13, and carbon-14. These all have the same number of protons, so by definition, carbon has 6 protons, but they have different number of neutrons, and that gives them a different mass. So carbon-12, that's the total mass of that isotope. And so an isotope by definition is an atom of the same kind that has a different mass. Some isotopes are stable, meaning they're in a configuration in which they aren't going to emit radiation. Some are radioactive, which means that they are going to emit radiation. So you can have stable isotopes and radioactive isotopes. Now, when those isotopes emit radiation, they will change their state from a radioactive to an active state. or from one radioactive state to another, but eventually they will decay, those radioactive isotopes, and as they give off radioactivity. And so one way to measure the amount of radioactivity in a substance or in a sample is the half-life, how much time it takes for one half of the atoms in a sample to decay, and this is going to be constant over time. Alright, so similar to isotopes, but now using a different subatomic particle, you can also have atoms that have different charges. Now what creates a charge is if you have a different number of protons and electrons. You can see that in this chart here. So if it has a charge, it's called an ion. So fluorine that has 9 protons and 9 electrons here does not have a charge, and so it's not an ion. It's at a stable or even a charge, or neutral. Fluoride that has 9 protons and 10 electrons now has one more negative than positive charge, so it has a total charge of -1. Anything with a negative charge is called an anion. Now it can have a -2 or -3 charge and still be an anion. Finally, you have some that don't have as many electrons as protons. So sodium with 11 protons and 10 electrons has a +1 charge, and that's called a cation. Okay, anions and cations are then attracted to each other and can form ionic bonds. And that's because opposites attract and because they want to form a stable charge. So together, if you have a cation and an anion, they can get together and create a neutral charge. All right, electrons have certain behaviors, certain paths in which they orbit the nucleus. So it's not actually round, as you can see in the electron shell diagram and some of the other ones we've already seen. They're actually shaped in a specific pattern. The Bohr model shows electrons found in certain areas circling the nucleus, and these are called orbitals. There's only 2 electrons per orbital. The s orbital is the first and most simple one, and it is spherical. Okay, so it does have kind of the circular but three-dimensional pattern for electron paths. The p orbitals are the next one, and they are dumbbell shaped. You can see those in this figure here. All right, so electrons, as they are floating around the nucleus, it takes energy to keep them there, and sometimes they actually jump to different energy levels. So these electrons then have energy, have potential energy, which relates to their position in an atom. As electrons move down an energy level, they give off energy. And in order to get an electron into a higher energy level, you have to input energy and cause them to jump. Okay, and this shifting of electrons in these different energy levels is also responsible for what happens when light or different colors of light are given off. That is because electrons are being excited, and then as they go back down to energy levels, they will give off light or energy in the form of light. All right, so the periodic table of elements, we have one of these in our large lecture room. It organizes elements according to a few things. One, according to their atomic number, and also according to their valence electrons. So you'll notice it's not just a list of 1, 2, 3, 4, 5. You also have kind of big gaps here and there. And the valence electron are those electrons that are on the outermost energy, outermost orbital. All of them want to have a full outer energy level, and this is called the octet rule. And so elements will form compounds, they will have reactions in order to empty their outer energy level or fill their outer energy level. both instances will cause their that outer valence electrons to either be lost or join with another atom. All right, so there are some elements are not equally represented in all living things. Most elements in living things are carbon, hydrogen, nitrogen, oxygen. So 96.3% of the elements in your body are these 4 elements. Many of them are also found in larger abundance relatively to others, and these are called minerals, and include sodium, chlorine, calcium, potassium, phosphorus, sulfur, iron, and magnesium. And then you also have some which are also vital for life, but are found in very, very small amounts, and these are called trace elements. An example of a trace element is iodine. Iodine is necessary for thyroid hormones to function. If you don't have enough iodine, what happens is your thyroid will actually swell, and it forms this large swelling in your neck called a goiter. Okay, so we kind of glossed over this before, but when atoms combine, they form different types of structures called compounds and molecules. A compound is a combination of elements in a fixed ratio. And an example of this is glucose. It has 6 carbons, 12 hydrogens, and 6 oxygens in a specific arrangement. And those elements are different, right? Molecules, on the other hand, are combinations of the same elements in a fixed ratio. So ozone has 3 oxygens, which have multiple bonds between them. Oxygen has 2 with a double bond between them, and you see that here. But they're both oxygen, so this is called a molecule of oxygen. You would call this a methane compound because it has carbon and hydrogen in it. Compounds and molecules have unique properties which are different than the individual properties of the atoms that they are made of. So CH4 acts differently than if you had them all separate. If you had 4 hydrogens and 1 carbon separated, they would have specific properties and be predictable in their behavior. But CH4 has a different set of behaviors. Okay, now elements and atoms are not static. They don't stay the same. They are actually constantly going through many different types of reactions, and these are called chemical reactions. What happens in a reaction is you form or break bonds between atoms or compounds or molecules, and those bonds change position. Some elements or atoms may be booted out of a compound or put back in, and there's many different ways in which that happens. So some of the simplest reactions we have listed here. In a chemical reaction, you have reactants on one side. This is the original atoms and compounds and molecules that you have. And then you have products on the other side. That's what happens after the reaction, what you're left with. Reactions are affected by many things, including the temperature. Generally, a increase in temperature increases the speed of a reaction or will cause a reaction to occur. The number of reactants or the number of products, and then a catalyst. And a catalyst is something that can help speed up a reaction without being part of it other than facilitating it. Okay, so it's not found in the reactants or the products, but is an important part of how it takes place. Some chemical reactions include oxidation reactions, and this happens when a molecule loses an electron, and generally those electrons are represented as hydrogen atoms. So and then a reduction is when a molecule gains a hydrogen atom. So in this reaction, you have chlorine molecule with the hydrogen molecule. They go through a reaction. That chlorine is being reduced because it now gains a hydrogen molecule. Oxidation, on the other hand, you have C2H6. This carbon loses hydrogen, so it is being oxidized in this reaction. All its hydrogens are lost to the oxygen, and so the oxygen then is being reduced in this reaction. All right, you also have bonds, which are formed as atoms are attracted to each other because they want to achieve a stable octet, right? So there are different ways in which this can occur. One way is if you have ionic bonds. So if you have two ions, such as a sodium ion and a chlorine atom that have a positive and negative charge, they can come together and form what's called an ionic bond. A covalent bond is where atoms are shared. So in an ionic bond, it's just that one has a positive overall charge, one has a negative overall charge, and then those two charges are attracted, and what holds those two atoms together. In a covalent bond, they actually have those electrons evenly shared, well, not necessarily evenly, but shared between them. In a nonpolar covalent bond, nonpolar means the electrons are shared evenly. So two hydrogen atoms that are sharing an electron, neither one of them has a greater attraction to that electron than the other, so they are shared evenly. In a polar covalent bond, those electrons are shared unevenly. So that occurs in water because oxygen is a bigger atom than hydrogen, and it has a greater affinity for that electron that they are sharing than the hydrogens do. So it's sharing, but it has it more than the hydrogens do. And the affinity of electrons is called electronegativity. So oxygen is more electronegative than hydrogen. Now, these negative and positive, or in a polar covalent bond, the compound will then have areas in which they have a negative and positive charge, because if it has a more electronegative element like the oxygen, it will have more of the negative charge there. And so sometimes between two atoms, you have a negative part and a positive part, and those form a bond called a hydrogen bond. You also have some bonds which occur between nonpolar compounds, and those are called van der Waals interactions, and those are actually what geckos have on the pads of their feet, which allow them to stick to different surfaces as they climb. Okay, so this is just a figure showing covalent bonds. Two hydrogen atoms are sharing two electrons here so that they can have a stable octet of two. Two oxygen atoms have to share 4 electrons, and so that's called a double bond. Two nitrogens have to share 6 electrons, and that's called a triple bond. So they each share 3. And these are all going to form a nonpolar covalent bond because the nitrogens are the same, and so it's evenly same in electronegativity, so they are evenly sharing those. Okay, so now we're going to talk about water. Water is an important molecule for life for many reasons, and most of them have to do with the ability of water to form hydrogen bonds very readily. Okay, and if you remember, hydrogen bonds form among these polar compounds. Now, the hydrogen bonds give water many properties, including cohesion. Cohesion is the ability of water to stick to itself. Okay, so if you know water if you watch water, it makes waves, it goes up and down, but that water always sticks to itself, and it can be separated from other water molecules, but generally sticks together, right? Adhesion then is the attraction of water molecules to other polar molecules, such as in these glass tubes. The water will actually climb up these glass tubes because of the hydrogen bonds which are forming between it and the glass. Okay, hydrogen bonds are also how some insects are able to walk on water, or which also be the reason why you might have water sticking to things like spider webs. All right, so this is a depiction then of those hydrogen bonds. The red is the oxygen, and it has more electronegativity than the white hydrogens, and so it's going to share, or actually kind of hog, those electrons and create a negative charge on the oxygen, while the hydrogens then will have a positive charge, and when those two polar opposites are in contact with each other, they will create a hydrogen bond. Okay? All right, so another thing that hydrogen bonds allow water to do is they give it a high specific heat. That means it takes a lot of energy to actually cause water to change from a liquid to a gas. Okay, and that process is called evaporation. Okay, we actually take advantage of this when we sweat. So when your body is overheating, you produce sweat. That way, when evaporation takes place, it actually takes energy in the form of heat away from your body and allows you to cool off. It also has a high heat of vaporization, which is the energy required to evaporate water. So these are kind of similar, and I kind of use them interchangeably here, but the specific heat or ability of water to absorb energy is because these hydrogen bonds are constantly being created and broken. That means it makes it hard for water to heat up, which then makes it hard for water to evaporate. Okay, so these are very similar in concept, but heat of vaporization actually is what has to do with evaporation itself. A kind of interesting phenomenon that doesn't occur in other liquid and solid comparisons of compounds is that ice, when it freezes, it is less dense than water. Now, everything else, when it becomes colder and colder and then starts to freeze, it actually becomes more dense and smaller. But ice, on the other hand, because of the crystalline structure that it forms as it gets colder and colder, with these hydrogen bonds becoming more and more static, it actually pushes them further apart. Whereas in water, they can get closer together and bump into each other. Okay, so it forms this crystalline structure, pushes them further apart, and thus ice floats on top of water because it's less dense. Okay. Now, again, because of these hydrogen bonds constantly forming and reforming, those bonds also allow water to be a great solvent, which means it will dissolve substances within it. Okay, pretty much anything that's polar or smaller things will dissolve and be embedded within or among the water molecules themselves. Water also opposes non-polar substances. So you can see that if you ever pour oil in water, the oil will rise to the top because it is non-polar, and those will not mix, at least not for very long. The terms for this are hydrophilic, which means it's water-loving. It will dissolve readily in water. Hydrophobic, does not dissolve in water, so it will rise to the top. Water also forms ions, okay? So it will break apart into hydrogen ions and hydroxide ions. And these are important for its ability to be a buffer. If you add acid or base, water can help kind of mediate those effects. All right, so this is a figure showing salt, which has a very rigid structure of a positive sodium and a negative chlorine packed tightly together. If you put that in water, these positive and negative ions will break apart because the positive and negative pieces of water will be attracted to those, and that's how things dissolve in water. Alright, so now we're going to switch to talk about acids and bases. Now, pH is a measurement of the concentration of hydrogen ions. So when we're talking about pH, we're talking about how much of these hydrogen ions are in a solution. It's measured on a logarithmic scale, which means if a pH of 1 has 10 times more than 0, pH of 2 has 10 times more than 1, and 100 times more than 0, pH of 3, 1,000, and so on and so forth. Water is neutral, meaning it has the same number of hydrogen ions as hydroxide ions, and that's pure water at least. It has a hydrogen concentration of 10 to the negative 7th mole per liter, and we will talk about what these calculations mean in class. It may not make sense right now, but we'll go into more detail in class. Acidic compounds have a pH less than 7, so anything 7 or below, 6, 5, 4, 3, 2, 1, those are all acidic. Basic compounds have a pH greater than 7. Alright, buffers resist a change in pH, so I just mentioned how water is generally a good buffer. There are other solutions which are even better at buffers. So a buffer will stay relatively the same in pH, and that's what you see on this graph here, even as you add more and more of a base or more and more of an acid. And that's because of their ability to absorb hydrogen or hydroxide ions. This range then is their buffering range, in which they can resist that change in pH. But eventually, if you add enough acid or if you add enough base, it will increase in acidity or decrease in acidity. Okay, so to calculate pH, this is the equation. It's the negative log of the concentration of hydrogen ions. Okay, and so that just gives you an easy number to work with. If you actually had the concentration of hydrogen ions, it's going to be this really large number, much harder to deal with. But if you turn it into this pH, it's just easier to do. The concentration is measured in moles per liter. 1 mole is 6.022 times 10 to the 23rd atoms. So that's a measurement of the actual number of atoms or molecules or compounds within a solution. A pH of 7 means there are 10 to the negative 7 moles per liter of hydrogen ions in a solution. Okay. All right, so why are acids and bases so important to life? Well, when a solution has a very high or low pH, it can be reactive. It can cause compounds to behave in a different way. They can also be very corrosive, able to dissolve and break down substances. So two examples which are used in common cleaners are Clorox and bleach and ammonia. Bleach is on one end of the spectrum, ammonia is on the other end, and they are principal component of many cleaning products because of their ability to break down substances. Hydrochloric acid, this is a biological example. This is stomach acid. It breaks down food. And other environmental effects caused by acid include acid rain, which is caused by human pollution, as is ocean acidification. We'll talk more about that in class. Okay, now we're going to switch and talk about chemicals of life. We are a carbon-based life form. You may have heard that before. And the reason why carbon is such a great molecule to use as a base for organic molecules is because it can make 4 covalent bonds with 4 different things. It has 4 outer electrons, and it wants 8. So it can share an electron with 4 different things. That allows it to bind with other carbon atoms while still being able to bind with 2 other things. So one carbon can be bound to 2 carbons and then have 2 other side groups which are completely different. Okay, this is a unique property which makes it unique for building, being a building block of life. It will also readily bond with oxygen, phosphorus, sulfur, nitrogen, and hydrogen. And also makes these hydrocarbons, okay, methane, ethane, propane, butane. These are just chains of carbons with hydrogens all around them. And these aren't organic, or these are not compounds found in life, but they are byproducts of some chemical, biochemical pathways. Okay? Now, the way that things are arranged on a molecule or on a compound also affects their properties. And so we call things that have the same molecular formula but differ in some way isomers. So we have structural isomers, and we have an example here. These actually have the same chemical formula. So you can count the 1, 2, 3, 4, 5 carbons. But the carbon is forming a chain in pentane, and in 2-methylbutane, it's branching off. Okay, so these are structural isomers. Their structure is different, but everything else is the same. You have cis-trans isomers. Again, they're the same. But in this instance, you have a carbon with a hydrogen and a carbon with something else bonded to it. And in a cis isomer, the 2 other things, or represented by an X here, are cis isomers, and when they're on the opposite end sides, they're called a trans isomer. All right, then finally you have stereoisomers, of which an example is an enantiomer. These are molecules that are a mirror image of each other. So an example of this would be these 2 structures, an L isomer and a D isomer. They look pretty much the same, but they are different just as the way your right hand and left hand are different. You can put your right hand and left hand together and they make a mirror image of each other, but you cannot put your right hand in the same direction over your left hand and have them be the same. Your thumb sticks out on opposite sides. So that's an enantiomer. These molecules that are able to make mirror images of each other are called chiral molecules or compounds. All right, so these carbon-based compounds allow for chains of carbons to be strung together. And how they are strung together, we can break up into 4 different types called macromolecules. Macro means big. These are larger chains of molecules which are important for life. Now, a more general term is called polymer. This means something that has a repeat unit. The unit that is repeating is called the monomer, and when you stick them all together, it makes a polymer. Now, polymers can be broken down into monomers, and monomers can be put together into polymers based on 2 different properties. The bonds between them can be broken by a process called hydrolysis. So bonds can be broken by hydrolysis. You see that, and that requires a water molecule because you need a hydrogen and OH on each of the new 2 ends of your now 2 new polymers. Or they can be made by a process called dehydration synthesis. So you form a bond and you have an excess H and OH, which form a water molecule. And the 4 different types of macromolecules are carbohydrates, nucleic acids, proteins, and lipids. So we'll go through each of those. But first, macromolecules have a common arrangement of compounds called groups. These are usually on the ends or the sides of these larger compounds, or even on the ends or the sides of the monomers within them. So these are ones that I want you to know. They're on here. Amino group is NH2, hydroxyl group OH, carbonyl group CO, carboxyl group COOH, sulfhydryl group, phosphate group, and methyl group. Okay, so you need to know these. And you will recognize them as you talk about different organic compounds. So carbohydrates are our first macromolecule we're going to talk about. They typically have a 1 to 2 to 1 ratio of carbon, hydrogen to oxygen. And that will repeat. But also, they are for storage, generally, of energy, especially in plants and animals. Starch is the storage molecule in plants. Glycogen is the storage in animals. They can also be structurally important. So, for example, there is a structure called cellulose, which is important for the cell walls of plants. These carbohydrates are made of, one example of carbohydrates are sugars. There are 3 different types of monosaccharides. That means they are one sugar by themselves. So you have fructose, glucose, galactose. There are more than this, but these are the 6 carbon ones. Okay? And here's an example of glucose here. It has 6 carbons, makes a ring structure. And you can put these together. They can form bonds between each other to make disaccharides, which means you have 2 of them together. So examples of those are lactose, which is found in milk, sucrose, which is found in which is table sugar. Then you have polysaccharides. These are 3 or more monosaccharides combined together. So glycogen, starch, cellulose, lignin, these are all polysaccharides. They have lots of sugars bound together. All right, nucleic acids then are another macromolecule. Their monomer is called a nucleotide. A nucleotide is made of a nitrogenous base, a phosphate group, and a 5-carbon sugar. And the sugar is either deoxyribose or ribose. If it's deoxyribose, then it's called deoxyribonucleic acid, or DNA. If it's ribose, then it's ribonucleic acid, or RNA. There are actually 5 different types of nitrogenous bases. 4 of them are found in DNA: adenine, thymine, guanine, and cytosine. And RNA has the same, except it does not have uracil. It does not have thymine. It has uracil instead. These are bound together through a bond called a phosphodiester bond. Okay. Now DNA, its purpose is to code for protein instructions. And so the sequence of bases, A, G, C, or T, is that code. It is that information. They are arranged in a double helix, which basically looks like a ladder that has been twisted. The hydrogen bonds are formed between these nitrogenous bases, and they are specific to certain types. So adenine will bond with thymine or uracil, and then guanine will bind to cytosine. So they are always paired in that order. There are 3 types of RNAs. You have messenger RNA, which is important for taking the instructions from the DNA to the protein. You have ribosomal RNA, which actually forms a ribosome, which then is an enzyme for making protein. And you have tRNA, or transfer RNA, which takes amino acids to the ribosome to have them stitched together to make a protein. We'll talk more about that later. There are other nucleotides, including adenine triphosphate, or ATP, which is the energy molecule. It's a very important molecule we'll talk about lots of times in this class. NADH and FADH2, these are intermediate energy storing molecules or electron carriers. important for photosynthesis and respiration. Okay, proteins are the third type of macromolecule. Their functions are a lot. They are essentially the functional part of living things. They are kind of what does all the work. Not necessarily all of it, but a lot of it. So they can be for enzymes, they can be defense, transport, support, motion, regulation, storage. All these things are very important, or fulfilled within the purposes of proteins. Their monomer is an amino acid. Okay, an amino acid, they are bound together by peptide bonds. Okay, you can see the diagram on the right, a peptide bond being formed. It has a central carbon, also called the alpha carbon, which bonds to a hydrogen, a carboxyl group, and an amino group. Okay, so these are two of the groups which we talked about before, and then an R group. And the R group is what defines the amino acid. All amino acids have those three things the same: the amino group, the hydrogen, and the carboxyl group. And it is the R group, or the side chain group, which is different. Okay, peptide bond, just like before, is formed by a dehydration synthesis between the amino group and then the carboxyl group of the next amino acid. So you can see you have three amino acids here on this figure. They have different R groups. One's yellow, one's green, one's smaller in green. The peptide bond is already formed between the one with the yellow R group and the large green R group, and then a new one is formed between the big R group and little R group one. Okay, but that is formed between the carboxyl group of one end and the amino group of the other. All right, so then this is just another figure showing the many different functions of proteins, which we'll probably go over in class as well. All right, so proteins have many different levels of structure and arrangement. The first one's called primary structure, and that is just the sequence of amino acids. So there are 20 different amino acids, and how they're arranged determines what they are, and we'll go over those in class as well. The secondary structure then, once you have that string of amino acids, are interactions between R groups within the chain, and they can form bonds, they can form large structures called like alpha helix or a beta sheet, and that's the secondary structure. The tertiary structure then is the final folded shape after all these secondary structures are put together. And then finally, a quaternary structure is interactions of multiple chains. So hemoglobin actually has 4 interacting subunits with the same or with different tertiary structure, which then come together to form hemoglobin. When an amino acid is made, it doesn't naturally have that tertiary and quaternary structure. So it needs help, and there are proteins called chaperones which actually help it fold correctly and be put in the right condition. So if this doesn't occur, this can be a problem. So which happens in cystic fibrosis, these proteins are not folded correctly. Now there are many conditions which will cause proteins to denature or unfold. Okay, so those secondary and tertiary structures basically fall apart. This may be because it's too hot or cold or acidic or basic or whatever. If it's not in conditions in which it can function, it will fall apart. All right, finally we have lipids. These are defined as nonpolar hydrophobic molecules. Okay, it includes fats, waxes, oils, and some vitamins and hormones, many of them which are cholesterol based. Okay, fats are one type of these. They have a glycerol backbone, which is attached to large strings of carbons, hydrocarbons called fatty acids. A fat then has 3 fatty acids on 1 glycerol. This is called a triglyceride. These fatty acids differ in structure. You may have heard of like omega-3 fatty acids or saturated fats or non-saturated fats. What happens for unsaturated and saturated fats is the number of double bonds. A double bond in a fatty acid makes an unsaturated fat, and you see that to the right. And what that does is that double bond messes up the ordered structure of the 3 fatty acids in the triglyceride, and so they no longer fit nicely together. Okay, if you don't have any double bonds, then they fit tightly together, and because of that arrangement, they are solid at room temperature. So fats are generally solid at room temperature. Oils are generally liquid at room temperature. Oils are unsaturated fats, and animal fats, butter and such, are saturated fats. All right, so finally, there are some lipids which are very important for the cellular components, especially in the lipid bilayer, which surrounds all cells. So these are formed by phospholipids. Phospholipids have a phosphorus head and a glycerol molecule, which then just acts as a connector. And that attaches to 2 hydrophobic lipid tails with usually a small kink in them. That phospholipid bilayer then forms a semi-permeable membrane by which some things are allowed through and some things are not. Another type of formation that can form from phospholipids is called a micelle, and you see that picture of it in the bottom right. Okay, that is the full video lectures for macromolecules of life. We will break those down in different classes and go into the different details within each of those classes.